So I think those were excellent talks that summarized the state of the field where we are now, and we're going to have a little bit of discussion here amongst the panelists before we open up questions to the audience. I'd actually like to start, if you will, with Beth. And you describe an intriguing mechanism and raise the question, how does environment converge with a genetic risk? Can you be explicit about how the mechanism that you've explored might serve as one potential locus for that interaction?
Yeah. So it's a really fundamental question, not just for this, but for autism and for other disorders you heard about today. But I think in our particular model in which we've identified this pruning mechanism that, as it turns out, involves this immune-related pathway. So one of the things that's, that we've thought about is one of the things we know is that these immune molecules and immune mechanisms are actually expressed in the brain, and they have normal roles. But just like in the immune system, they're highly regulated by the environment. So stress, infection, inflammation, you can name many, many different possible perturbations that in the immune system we know leads to a dramatic change in the expression, function of these molecules. So our work and others have shown that complement, for example, is highly regulated by inflammation, inflammatory signals, and even stress-related signals. Now, the idea there is, if you think about the fact that what I've described today is an immune-related pathway that normally helps prune synapses, but then it turns off after this normal critical period. And that's good. You don't want to have this pruning continue on. But you can envision a scenario where too much pruning, because either the brakes are gone or some other perturbation, whether it be genetic, environment, or both, lead to too much of a good thing. And that's what we envision might be happening in the context of schizophrenia, but also perhaps in other disorders as well. And so the idea is we can actually start to think about modeling this. Now, there are no good animal models of schizophrenia, I believe. However, the genetics are enabling us to start to think about creative new ways to model this. In mice, to some extent, we can put overexpression of the human version of A, for example, and see how that impacts circuits. We're doing that now. This is actually work by Mike Carroll and in collaboration with Steve McCarroll and I. But we're also imagining doing this in non-human primate models. And we're even starting to think about organoid and human-based stem cell models through multiple ways to test the idea that too much complement leads to too much synapse loss. And then you can give a second challenge where you perturb the system, either with a stressor or an inflammatory challenge, for example, and see if that exasperates the problem. So we're starting to get at that. But there is some basis for the idea that environment normally does affect these pathways under normal conditions. By the way, it also really dramatically impacts microglia, which are the immune cells that are sort of downstream from this process. So now the key and the challenge ahead of us is to start to think about how we can best model this and then relate this to the human data. And I think that's where we just need better model systems, and that's what we're actively working on at the Stanley Center and with our collaborators.
So it's really exciting that you can point to this mechanism and at least start to get down to some fundamental biology,
right,
with the hope that that will translate back upwards as well.
Right.
Matt, Chuck, where are we in terms of mechanistic applications in these earlier neurodevelopmental disorders like autism and neglect syndromes?
So where are we? Well, we're a lot better off than we were before gene discovery. So on the one hand, I think that there are several things I think that are converging that I think really give us the opportunity to be optimistic about progress and understanding mechanisms. So first is that we clearly have signal that's pointing to different biological mechanisms. There's a lot left to explore there. We sort of just touched the surface in terms of understanding critical role for the synapse, as I said, and for chromatin modification. There are other more specific signals around things like RNA binding proteins and fragile X mental retardation protein. So there's a lot of work going on there, I think, to try to begin to disentangle. One of the reasons that I showed the slides that sort of asked a slightly different question is that I think, to think about when and where this risk might be emerging is that I think for model systems, they can be tremendously useful, but being able to constrain model systems in a way that begins to sort of narrow in on when and where you might want to look to get a handle, not just on biology, but to really begin to kind of triangulate on pathophysiology. And I think that's starting to happen now. in a way that you can imagine this moving forward to elaborate early mechanisms. So I think overall the news is good. I wish we were further along than we are, but when you think about the rate of change over the last 5 to 10 years, always look to cancer and other disorders where the ability to begin to get to molecular mechanisms really transformed the lives of children and adults with these disorders. And I think, I don't know how long it's going to take for autism, but you have to have the same hope that this kind of insight is a harbinger to really something tangibly important.
So I'm going to switch gears now and talk about my life as an autism researcher. Towards the end of Matt's talk, he showed a slide where he pointed to this sort of late perinatal, early prenatal inflection point. In our work looking at infants at high risk for developing autism by virtue of having an older sibling or two, where the prevalence then is 20 or 30%, we find that we see at 3 months of an age a distinct signature in the EEG pattern that predicts autism. And so if we look at the high-risk sample, those with siblings, versus a low-risk sample, we see a difference. But if we then divide out the high risk into those who then develop autism versus do not, this signal is present in those who do at 3 months. So it's occurring at around this point. There's a couple of issues that we need to sort of take apart here. One is, if that's the case, autism is developing much sooner than we think. And we probably should not be thinking of this as a postnatal disorder as much as a maybe prenatal, early postnatal. The second, going back to something Beth said, is the issue of biomarkers. So we study more than 6 rare genetic disorders, all of whom increase the risk for autism. And we do that because in the infant SIB work, we know we're predicting autism, but do we know that it's autism specific? What if we're simply predicting a brain that's gone awry? Mm-hmm. And keep in mind that early in development, there's a lot of dynamic and flexible processes. So a genetic hit or an environmental hit can simply shift the balance in one direction or another. But predicting where the ball eventually rolls is much more difficult. So what we need to reassure ourselves of is that we're predicting autism and not simply an atypical pattern of brain development. On the practical side, then the question is, well, if we have this tool, should we be using this clinically? And I think it's early days for that because we can't make a mistake. If we have type 1 and false positives and false negatives, the last thing we want to do is tell a parent of a 3-month-old, we're sure your baby is going to develop autism, or that your baby will not develop autism, and be wrong. So we have a lot of work to do. And the last thing mechanistically, to bring this back to where you started, is that with my colleague Takao Hench, or our colleague Takao Hench, we're working on models of excitatory inhibitory balance. And the idea is that what we're seeing in the EEG in the human is similar to what he sees in the mouse. And the question is, if we shift the excitatory inhibitory balance, can we bring things back into alignment?
Deanna, Chuck raised the issue of prediction early in autism, and you talked about some evidence, particularly in the motor sphere, that there are detectable precedents of the development of schizophrenia. How early can prediction go in schizophrenia?
So the best domain is motor, and that's in infancy. So you can look at some of the really interesting studies were done by Elaine Walker at Emory, where she actually got parents' videos of their kids, both of the kids who went on to develop schizophrenia and the kids who didn't, and you can see these early neurological soft signs as early as infancy. And there's some predictive utility, but again, we're not at the point where we necessarily want to go in and say, we think your child's at risk for developing schizophrenia, because it's not that strong of a prediction, and unfortunately, we don't have an intervention that we can offer at this point in time.
So, is schizophrenia happening at the same time? Is it so early? Is it there already in mid-fetal development like we think autism is? Please.
So I would say at this point, we do not know so 2 very simple hypotheses that I think are potentially testable over time. And that is, one possibility is that there's some subtle, early, even prenatal defect, much like autism, that is actually there and affecting synapses and circuits in very subtle ways early on. And it's not until later in development, during, let's say, the second wave and this later adolescent critical period, that it becomes exposed. Now, that could be that, some other hit, or the fact that that's a very dynamic time in those particular circuits that could then unveil the disorder. And that's sort of what we're thinking might be possible. Another possibility, though, is that there is less of a change early, and then it's all happening at that time. And so for us to really tease that out, we need to have access to human brains and samples where we can look over time and do a longitudinal, which of course is very difficult to do. And we're starting now using brain samples that are available over time to try to get a sense of when things start to change. But I think that's been the challenge in schizophrenia. The image I showed you from David Lewis, I mean, these are adult samples where you see decreases in synapses. And even with the imaging experiments, those are done, much later after the onset of symptoms. So we don't really have much data on at least looking at the brain, looking at structure, and looking at mechanism. We just don't know. So I think with some of the animal models we were describing, the fact that we have genetics that are enabling us to start to model some of this, we may start to be able to tease that out. But I think it's a hard one to address in humans. I don't know if anyone else has comments to add to that.
So just riffing off that a little bit, I guess one of the things I'd like to sort of point out is that this idea that there's a convergence of risk in autism in mid-fetal development doesn't mean that the game is over in mid-fetal development. And already you already heard from Chuck that we see an unfolding of symptoms in high-risk families. We see, surprisingly, that some of the earliest markers of autism, which is gaze preference or differences in gaze preference, actually start out the same in infants as far as we can tell. And it's over the first 6 months of life they begin to diverge. So I'm not certain that they're not very similar stories with sort of different time windows occurring. And I think it's very important, particularly when I talk about this in autism, that it's easy to kind of get into this kind of therapeutic nihilism if you think that everything bad is happening in mid-fetal development. But in fact, knowing essentially nothing about the basic mechanisms, we also know that kids with autism, with early intervention do better than without. So we see continued plasticity, even after the onset of illness. I just wanted to.
Go ahead, Deanna.
Well, just to also bring it back to this important interplay between the genetics and the environment as well. So if you think in schizophrenia, what's interesting there is that there is this evidence, population-based evidence about environmental risk factors that happen around the time of, second trimester that often impinge upon the immune system. And you might see then that actually shapes the child in a way that they react to the ongoing environment in a different way. So unlike depression, where there's a lot of evidence that life events actually precede the onset, there isn't in schizophrenia, but it may be the child's reaction to life events that's been shaped by the genes and the environment that lead to this adolescent sort of specific period of risk.
So I want to switch gears for a moment and bring up a topic that I raised in the introduction, which some of you have touched on, but which we really focused on, which is heterogeneity. I want to start with you, Chuck, about what in the studies in the Romanian orphans that you mentioned. How much heterogeneity is there in the phenotype that comes out, and can we use that to get an understanding, with a single cause? On a complex genetic background, what kind of heterogeneity can we result?
So that's a great question. And so, clearly we have major group effects across multiple domains, but when you do a deep dive in there, you see a lot of variability. So for example, there may be a child who has spent his or her entire life in an institution, but IQ has been relatively spared. So instead of having an IQ of 60, they have an IQ of 100. On the other hand, they have tremendous problems with mental health. They have high callous unemotional traits, ADHD and the like. So where we need to be careful is that we see a lot of heterogeneity in outcome, and we can explain some of this genetically, and we can explain some in terms of individual differences at a more superficial level. But the challenge is, it's not as though there is resilience or sparing across the board. What we see is resilience or sparing in some domains, but perhaps not all domains. And that's important to emphasize. It's not as though a child removed from the institution and put into a good family shows complete recovery, or a child who's never left the institution is across the board impaired. We have to look at these domains, and we know in critical periods there's cascading critical periods. So we probably shouldn't be surprised that some domains respond more to the environment than others, and perhaps because of genetic breaks, some may respond less to the environment than others.
Deanna, you said something that suggested that at least at the cognitive level, the heterogeneity may have more to do with genetic background than individual experience differences. In that you said, that if you look compared to the expectations from where their parents would be, most if not all individuals end up lower. How much variability is there in that sphere when you, if you will, control for genetic background looking at parents?
Well, if you control the genetic background, then there's actually more homogeneity, right? Because it looks like that you've got, so if you don't control for that, it looks like there's a lot of variability all the way from people who seem to be relatively intact versus impaired. But if you think of it as relative to where they would have been, then it seems to be that there's more of a, like some people have estimated 91% of individuals with schizophrenia have cognitive impairment relative to what you would have expected given their genetic background.
So what do the genetics then tell us about heterogeneity, Matt?
In autism? Well, the first thing is we see the same thing in autism, and there's this question about this, that, how much are we seeing with regard to intellectual disability versus social disability? Right. But the story is very, very similar, right? So I think there's an interesting kind of dichotomy. We know that autism is tremendously heterogeneous, and I had a table that showed 65 genes, and none of those genes account for more than 1%. In fact, almost all of them account for far less than 1%.
But within one particular genetic cause, where you have the most, most accumulation of one, is it more homogeneous when you have a particular genetic mutation?
That's a great question. I think overall, it's likely that there are constraints. You can begin to see, we take one of the top genes, CHD8, and begin to look, and by and large, they're more likely to have slightly larger heads. They're more likely to have a particular IQ profile. So there is a bit more of homogeneity, but really, it's not as though it becomes highly specific. But I'd say the flip side, which is important from a clinical standpoint, is that it's enormously heterogeneous. At the same time, the clinical diagnoses that have been around for 40 years are enormously good. Like, that's what we relied on largely to do gene discovery. And so it opens the door. So you can see kind of this iterative process. You have a lot of heterogeneity, but enough convergence that it leads you to begin to identify, biological markers. And I think the question then about the genotype or the biological phenotype relationships are really going to begin to be clarified.
Okay. Well, thank you very much for the panelist discussion. We now would like to open up the panel to questions from the audience. Please come up to use the microphones here on the aisles. Please go right ahead.
Betty Sue Masters from Duke University. I have a question of Dr. State. Are there any data relating to IVF children? Addressing your point about pre-fertilization and the genetic defects. And then I have another question of the audience, but go ahead, please.
Yeah. So I was going to say there's been some interest in IVF. I don't know of any good population-based study that demonstrates a clear increase in risk. But
right. If you look at assisted reproduction, generally it increases the risk for a number of things, but nothing that's specific to autism.
Okay. And then the other question I had was about the development of genius and specific skills. Could you address that?
We have a genius here, I think.
Don't ask me.
Now, I have to apologize for saying that.
I know, but it's a great turf. I definitely want to turf it to. I have. So it is the case that I think that the notion that there are individuals with autism who have savant skills is clearly true and is supported by the data. There's essentially no understanding of that, and as far as I know, no genetic studies that have looked at that group or that population.
And the same goes in the neuroimaging domain. I mean, you might see a few little differences in one area or another, but there's nothing in the anatomy of the brain that would predict who's a genius.
Here, we're going to go over here to the other side for a question.
Jim Perrin, Mass General and Harvard. Just want to continue on the discussion of heterogeneity here. So the advantage of the Simon simplex is it's a relatively homogeneous population. I say relatively because it's not extremely, but when you think about application of some of the new genetic findings to the very heterogeneous variety of disorders, what do you expect we're going to see? And of course, wasn't there that study a couple of years ago that showed that siblings, both of whom had autism, had quite different genetic patterns?
Thank you for the question. There's a lot in there. So the first thing I'd say is that there's always an ascertainment issue when you do a clinically based study. These are not epidemiologically derived population samples. At the same time, I think that there may be kind of an overestimation of how homogeneous the Simon simplex collection was. Its IQ range from moderately, severely intellectually disabled all the way to IQs of 160. These were families that were really identified to sort of be quote unquote guardian garden variety autism cases. So you would say that the kids that would normally make it to a medical geneticist likely were not in that group, at least over-enriched in that group, but I think that they probably were pretty representative of what you'd see. And there, what we can tell you is that, about 10% now of kids that present that way with quote-unquote idiopathic autism, you would expect with some genetic testing to identify a de novo event. The kids who see medical geneticists with intellectual disability, seizure, some dysmorphology, et cetera, the rates of identifying a particular cause of autism much higher, maybe as high as 50% or so. So I think it's beginning to sort of infiltrate at least potentially the clinical realm. I think the biggest question for us is that the lack of specificity makes it very tough, particularly for those idiopathic kids that, you can find something, but most of what we're going to find, we're just going to tell the parents, we think that this we know it increases the risk, but we can't tell you exactly for which disorder, and it's possible that there'll be no serious outcome. So we have a long way to go, I think, from a predictive standpoint to leverage the current genetic findings.
The only thing I'd add, Jim, in terms of heterogeneity is that if you look at the large number of populations that have increased risk for autism, they themselves are heterogeneous. So Duchenne muscular dystrophy, Down syndrome, TSC, Rett syndrome, and the like, 5 to 10% of the kids who grow up in institutions have autism. Whether this is the same disorder, of course, clinically it may be, but mechanistically, I would be very surprised if we're talking about the same underlying mechanisms that lead to such that follow such disparate pathways.
Back over here. And I was supposed to remind people to keep their questions brief, although everyone's doing such a great job of it so far, it may be unnecessary. Go ahead.
I'm Bill Hazard. I'm from Wake Forest University, and my interest has been in aging for a long time. However, I believe that begins at conception and ends at death or maybe sometime afterwards. But I learned at Cornell as a medical student that schizophrenia was called dementia precox. So what I would like you to comment on, though, is a more finely nuanced description of the adolescent process, when it begins, when it's altered in its development, especially by gender, and how that may influence the expression of these underlying proclivities.
Anna, you want to take a stab?
You don't want to lie. I think it's interesting because there's heterogeneity there as well, right?
Yeah.
So we see some people who will end up with a diagnosis of schizophrenia where you do see what was referred to the kind of the dementia praecox process, where you see this decline. They were maybe doing well in school, had friends, and then, early adolescence, you start to see, they're doing poorer in school, they're pulling away from friends. It's not clear, and that there's some evidence that those folks will actually start to show a decline in cognitive function as well. And then they'll start, with the manifest symptoms later on in adolescence and end up with a full-blown diagnosis by late adolescence, early adulthood. So that's really what was captured by that idea of dementia praecox. And that is certainly a subset of the individuals with schizophrenia, but that's not the entire set. There's another set of a more acute onset where maybe they didn't have really clear premorbid impairments. There wasn't this period of decline over time, but there'll be sort of more out of the blue onset of particularly like hallucinations and delusions. And we have not, unfortunately, really identified clear genetic differences between these subgroups to understand why you get in some people this decline over time, which coined that term, versus the people who have this more acute onset. The folks with the more acute onset do seem to have some better outcomes. They are, on the average, more likely to have a higher ratio of females in those, although the females are not represented in the other group as well. We also have a lot of interesting information about gender differences, so there tends to be a later age of onset for women. They tend to have a better response to treatment, although that may be partly because the families are more likely to support them. There may be interesting hormonal effects having to do with some of those gender differences as well.
Thank you.
Corey.
Anna partly answered my question, but I was just struck listening with this idea of iteration and returning to some of the observations, epidemiological observations. Matt brought up the second trimester, and that made me think of the Dutch hunger winter and the later onset of neurodevelopmental disorders. And I was remembering some things about metabolites and things that people were measuring, in the 60s in various psychiatric patients, and wondering whether some of those ideas, again, that had been discarded because they were very heterogeneous and who knows what they meant. If some of those ideas had been reevaluated with our new understanding of the heterogeneity of these disorders.
I mean, I think it's
sure. Yeah, I mean, I would take a stab. Yeah, I would take a stab at the environmental causes, and I think there is definitely renewed interest at evaluating the environmental causes, both from a mechanistic standpoint by doing things like Beth suggested, which is combining genetic and environmental models and seeing if you can exacerbate it, but also from an epidemiologic perspective and revisiting that. I know that the National Institute for Environmental Health Sciences has a program in trying to look at some of the environmental influences in early development and assess their effects later in life. There are a number of cohort studies that are ongoing. So I would say there is renewed interest, but not yet progress. I don't know if anyone would want to add to that. Okay. Back over here then.
John Morrison from UC Davis. My question is primarily for Beth Stevens. So the hypothesis has been put forward that the synaptic pruning may reemerge pathologically with cognitive aging and Alzheimer's disease. Can you comment on that?
Sure, absolutely. So work from our group and from others now in the field have suggested that there may be common pathways that also could help explain synapse loss across other disorders, including diseases of the age brain, including neurodegeneration. And it turns out, in the case of Alzheimer's, as you mentioned, there is evidence to suggest, both on the genetic standpoint, genes linked to microglia and complement, as well as evidence of synapse loss that happens quite early in Alzheimer's disease. And actually, many of the mechanisms I described very briefly today, we find in animal models and some evidence in human brain that this pathway may be aberrantly reactivated in the adult brain to contribute to synapse loss and cognitive dysfunction. Now, this is kind of an intriguing idea, but one concept is that although Alzheimer's and schizophrenia are so very different with respect to their age of onset, the potential, the genetics, the timing, it could be that this converging mechanism that then converges onto this complement or this pruning pathway. And if that's true, I think this is exciting and it offers us possibility for therapeutic intervention and potential new biomarkers that if we could figure that out in Alzheimer's where it's a little bit more tractable, this might someday be able to be applied to a much less tractable and more challenging neurodevelopmental and neuropsychiatric disorders. And that's the way we're thinking about it. So much of the other half of my lab is on neurodegeneration and aging, and we've made a lot more traction in this case. And I'd say the human genetics are also paving the way there because almost half of the genes increasing risk of Alzheimer's are either expressed exclusively in microglia or enriched in these cells, as well as a number of immune mechanisms. So that is sort of what we're hoping is that over the time that we're digging into that, it may provide new insight into schizophrenia and other disorders in the developing brain.
So just a heads up, it looks like we have 4 people left. We probably have about time for that many questions. We'll see if we can get to any additional ones, but we'll certainly get to the 4 here on the left side.
Thank you. Lin Gerber, George Mason University and Inova Health System. I have a quick question for Dr. Stevens. Do we know of any natural experiments of complement deficient either children or adults who do not have schizophrenia, or is there a mouse model in which you look at pruning effects with complement deficiencies?
Well, first of all, I can say that the C4 story is one of many genes and one of mechanisms, one of many mechanisms in schizophrenia. I think that increases the already 1% risk to about 10%. So it's still only one of many genes, just to put that out there. But I can tell you that we have been quite interested, and people often ask me, especially when I'm in a meeting like this, what do we know about people that are deficient in complement? Are there any cognitive impairments or any links to other diseases? Well, it turns out there's lots of complement pathways there's some redundancy there. But one of the things we know are people that lack C1q, for example, and other complement genes, they have more increase to autoimmunity. And there's been much less looked at in terms of cognition and developmental in those patients. But there could very well be redundancy because C1q, for example, is one of many different ways you could activate the complement cascade. However, that said, we are now collaborating and trying to gain access to patients that lack or have deficiencies in complement to start to put this together. It just the sample sizes haven't been enough that we can start to make any conclusions yet. But I think it's an excellent point.
Hi, Alison Goat, Mount Sinai, New York. So I think my question is probably for Matt, but anyone's welcome to answer. So I think it's pretty clear that both autism and schizophrenia are really highly polygenic disorders. And from GWAS studies, we can obviously now calculate polygenic risk scores. So I wonder whether you could comment on looking at those polygenic risk scores in the normal population. If you have a high polygenic risk score for either of these disorders but haven't developed it, what traits do we find associated with that risk score, and can we learn something about resilience from these people?
Yes, I will. Great question. So polygenic risk score is a way of taking a look at sort of the entire complement, no pun intended, of genetic risk, particularly common genetic polymorphisms in an individual, and essentially summing those up to give you a sense about how much risk there is. And that can be done either by identifying genes where you know that they're individually associated and then seeing how many changes in those genes an individual has, or it can also just be inferred from the entire genome, even without looking at specific genes that carry risk. So there are two different stories here. In schizophrenia, polygenic risk scores turned out to be a highly tractable way of thinking about identifying groups that are at very high risk. This is in part because there are 108 loci, actually more than that now, something like 150 or more, that have been identified where you can get a polygenic risk score that's shown to account for a significant amount of the variance. And that, I think, will be a very powerful tool to bring to studies, epidemiological studies, studies of neuroimaging, of treatment response, et cetera, to try to get a broader sense about the total genetic load in a way that is clinically and relevant to the population. With autism, it's been a different story. We can infer that the majority of risk for autism is carried in common variants, but the effect sizes are so small that we haven't yet reached samples that allow us either to identify a single common polymorphism that is reliably associated with autism, and our polygenic risk scores account for almost none of the variants. If we get much larger samples, then we should be able to do the same kind of thing in autism that we're now beginning to do in schizophrenia.
Oh, sorry, Deanna,
Just
go ahead.
follow up on that. I think one of Allison's points, too, is if you look in the PRS studies in schizophrenia, they are converging on traits in the general population that map onto the things that have, so you see it related to cognitive function, and there's starting to be evidence that it's related to cortical thinning, for example, in some of the similar areas. So they are some evidence that they're converging with risk factors that have been identified through other pathways as well.
Maureen Lichtveld, Tulane University. Back to the environmental factors and to the environment in general. Our lab studies the interaction of chemical and non-chemical stressors on early neurodevelopment. And so we know the lead and mercury and organophosphate pesticides, but particularly on the non-chemical stressors, prenatal stress is known. But my question is, in connection both to the Bucharest study and the study that Dr. Stevens has done, is looking at not necessarily only the individual, but the setting, and particularly community safety and community violence, and the influence on early neurodevelopment.
So I'll have to change set for a moment. I run a very large project in Dhaka, Bangladesh, looking at kids growing up in horrific circumstances. And we have lots of information about their psychosocial environment, and we have every biological specimen you can think of. And now what we're trying to do with Beth's help, although she doesn't know this yet, is to try to integrate these data sets to some degree. We find that the inflammatory markers, for example, and some of the other biomarkers we have selectively account for some outcomes, and the psychosocial, another set. But it's probably that interaction that's most concerning. So the mom with an untreated depression, or when there's domestic violence in the home, and the child is malnourished, and they have unclean water, and the like. And I think one thing that has only been implicit here is, sort of the viral hypothesis, the inflammation hypothesis of schizophrenia and autism has been around for many, many years. So what we need to do is model how these environmental factors collectively impact development. But what we're not doing too much yet is the genetic background, which has to play an enormous role as well.
Such a key thing, the genetic background.
I'm Godfrey Oakley from Emory, and my question relates to thalidomide. Marilyn Miller from Chicago pediatric ophthalmologist examined 100 kids for a weird and wonderful eye abnormality and found that 5 of those kids had autism. And I just wonder, a lot's known about the biology of thalidomide, and I wonder how that biology and the genetic biology might be crossing over, or have people looked at it?
Well, I don't know if I can answer the question the way you asked it, but I will say that 5 of, what was that, 100?
5 out of 100. I mean, it's an amazing attack rate.
It's above chance, but we don't know. It's a select sample, so those 5 would have had autism even without exposure to thalidomide. So, but mechanistically, I'm wondering, maybe you two can, yeah. Sorry.
All right, thanks.
All right. Well, thank you very much for the audience for the questions. I think the panel has been really outstanding, and I don't have much in the way to close it. So.